The nucleus is a cellular organelle found in eukaryotic cells that contains most of the cell's genetic material in the form of DNA. DNA contains the genes, which are essentially blueprints for various proteins that the cell needs to live. And most cells in the human body have a single nucleus. But some cells, like red blood cells, have no nuclei, whereas others, like skeletal muscle and liver cells, have more than one nucleus. Now, the nucleus is surrounded by the nuclear envelope. Inside the nucleus, there's the nucleoplasm, which is a liquid environment very similar to the cell's cytoplasm. Suspended in the nucleoplasm, there's chromatin, which is the densely packed DNA, and also the nucleolus, which makes ribosomes, which in turn helps build proteins. Let's start with the nuclear envelope, which has an inner and outer membrane, both of which are made of phospholipid bilayers. The outer membrane has lots of anchoring proteins that allow the nucleus to remain suspended within the cytoplasm, like a puppet on strings. The inner membrane is covered by the nuclear lamina, which is a network of lamin proteins. These lamin proteins are thin filamentous proteins that create a dense protein web within the nucleus, kind of like a dense spider web. The nuclear lamina provides something for the chromatin to drape itself over, kind of like caterpillars hanging out all over those spider webs. The nuclear envelope is selectively permeable, meaning it allows some things to pass through, while preventing others. There are also relatively large nuclear pores, and each pore has a nuclear pore complex lining it on the inside, made out of proteins called nucleoporins, and it kinda resembles a basketball hoop with a net. So large molecules like nucleic acids and proteins aren't able to come and go easily, but small water-soluble molecules have no trouble at all. The main role of the nucleus is to house the DNA. It's essentially a central genetic library with instructions on how, when, and what kind of proteins the cell needs to make in order to live and perform its functions. These instructions come from one of our DNA molecules that are really, really long, over 2 meters each when fully stretched. So our cells have to rely on a few packing tricks to compress all that to fit inside a nucleus. Here's where chromatin comes in. Essentially, chromatin is a fine matrix of very densely woven and compressed DNA. There are 2 types of chromatin. First, there's euchromatin, which is loosely packed and contains genes that the cell frequently transcribes and translates. In other words, euchromatin contains the genes that the cell frequently needs to copy from DNA into messenger RNA, or mRNA, and then into a protein. Second, there's heterochromatin, which is densely packed and contains genes that the cell rarely transcribes and translates. Now, chromatin is actually made out of 46 separate DNA molecules, each of which is called a chromosome. Most of the time, the cell's not dividing, and the chromosomes are in an intertwined, loose spaghetti-like state. During cellular division, the 46 chromosomes condense and double up the amount of DNA within the nucleus by creating an identical copy each, now called sister chromatids. So while there's still 46 of them, they're made out of 2 chromatids each, and take on that familiar X shape. The 2 sister chromatids are identical to each other and to the original genetic template, and they join together in the center in a region called the centromere. But they still make up a single chromosome. After the cell divides, each resulting daughter cell gets 1 copy of the genetic material. So 46 chromosomes made up of a single, loose spaghetti-like chromatid thread each, now called the chromosome again. And we're back to regular cell business. It can be kinda confusing, because the structure with 2 sister chromatids connected together is called a chromosome. And when the chromatids split up, the structure with a single chromatid is also called a chromosome. Now let's focus on the chromatin itself. It's actually a long, loopy, and continuous fiber. Zooming further in, the fiber actually resembles the string-like cord of an old telephone, so a tight helical fiber. Going even deeper, the cord itself is made up of tight loops of DNA wrapped around protein structures called histones. Basically, 8 histones get packed together in 4 stacks of 2, and the double helix DNA wraps around them twice. Together, these 8 histones and the DNA wrapped around them are called a nucleosome, and they look kinda like beads on a string. Now, the histones help control the DNA that runs around through them. If the histones have a methyl group attached to them, they prevent that chunk of DNA from getting transcribed. Finally, we have the nucleolus, which contains a special kind of DNA called ribosomal DNA, or rDNA, which can be transcribed into ribosomal RNA, or rRNA. rRNA is then folded around some proteins inside the nucleolus to create a molecular machine called the ribosome. The ribosome can float out of the nucleus, where it joins other ribosomes in the cytoplasm. Interestingly enough, the assembled ribosome is the largest structure that can pass through a nuclear pore. The ribosomes can then use the mRNA copies to produce proteins. The ribosome converts mRNA into a string of amino acids, which can form a protein in a process called translation. As it progresses, it'll inject the protein into an organelle known as the endoplasmic reticulum. Inside, the protein gets folded into shape and modified as needed to take its role inside or outside of the cell. Alright, as a quick recap. The nucleus is surrounded by the nuclear envelope, which has an inner and an outer membrane. Nuclear pores run through the envelope, and they control the kind and flow of molecules that are allowed in and out of the nucleus. Inside the nucleus, there's DNA, which is tightly packed into chromatin. Inside the nucleus, there's also the nucleolus, which assembles protein-making ribosomes.